Thank you very much for introducing and for inviting me. I'm going to talk about rare diseases from my perspective as a child neurologist who sees a lot of rare diseases. And as we have heard, rare diseases may be individually rare, but they are very common as a group. And there is a huge discrepancy between the substantial individual and societal disease burden and the lack of effective cures for these conditions. Rare diseases are particularly common in pediatrics and even more so in pediatric neurology. And I've broken down the almost 600 patients we see in the neuromuscular service at the Evelina Children's Hospital, my hospital. And the two observations which are striking is, if you look at the pie chart on the left, almost 95% of conditions we see in this service are genetic and only a small proportion are acquired. And what is also striking, that amongst the genetically resolved conditions, which are shown in the pie chart on the right, actually all of these conditions are rare diseases. And although these conditions are individually rare in a specialist service, we see a large number of these conditions. For example, the most common condition we see is Duchenne muscular dystrophy, which has a frequency of 1 in 3,500, but we have almost 100 patients with this condition in our service. And that gives us the opportunity to characterize these conditions very deeply on a clinical level and applying other approaches such as imaging, histopathology, and genetic methodologies, which provide the basis to understand the basis and the mechanism behind these conditions, but also to define outcome measures, to define natural history as a basis for future clinical trials. And I just wanted to give you an idea about current treatments, in particular in neuromuscular early onset rare diseases. We have less than 10% of conditions where we have treatments. The most effective treatments we have at the moment are for spinal muscular atrophy. However, these genetic therapies are prohibitively expensive. They do not cure the condition, so there is certainly space for other drugs, including repurposed drugs. Another example is X-linked myotubular myopathy, which is an ultra-rare condition with less than 1 in 50,000 boys affected. There is a gene therapy which was very effective in animal models, improved clinical outcomes in affected boys, but which also had very severe side effects, which was fatal in 3 cases due to liver failure. And that emphasizes the importance of understanding the natural history of rare diseases, but also the importance of considering repurposed drugs where the safety profile is better understood. I also want to highlight Duchenne muscular dystrophy as a condition where a repurposed drug, tamoxifen, has been used already. However, although there was a trend to improvement, that was not statistically significant. So there are huge opportunities in pediatric neurology rare diseases, because with the introduction of next generation sequencing over the last years, we increasingly understand the molecular basis and the mechanism underlying these conditions. But at the same time, their number is small. These conditions are phenotypically very diverse, and that results in challenges understanding their natural history, coming up with effective trial design, and in a lack of industry interest. And I want to challenge in the rest of my talk, in particular the last point, by emphasizing that rare diseases have probably closer associations with more common problems, and that therefore drug development and drug repurposing in rare diseases may also be beneficial for much more common conditions. And to illustrate that, I want to describe a group of conditions I have been working on for the last 10 years. And that's research which was prompted by a single patient I met 15 years ago, who was a very severely affected boy who had severe multi-system involvement affecting the brain, the hearing, the eyes, the heart, and the immune system. And he sadly died at the age of 3 months. And there was a situation, he had a condition called Vichy syndrome, and at the time only 8 patients had been described. So there was a very severe condition with a strong need for therapies, but because of the rarity of the condition, nothing was available at the time. So to take that further, we started an international collaboration, and based on a group of 10 patients, we identified the underlying gene defect, and the gene identified in this condition, EPG5, was not very well annotated at the time, but had been previously implicated in breast cancer, and very recently, before our paper came out, was associated with a very essential biological pathway called autophagy. And what autophagy does is to target intracellular material through specialized structures, autophagosomes, and directs these structures to the lysosome for digestion and recycling. And that mechanism has very fundamental roles, both in cellular metabolism, in infection defense, but also in ensuring the quality control of proteins and organelles, and is therefore particularly important in counteracting the effects of aging. And before we identified autophagy as the defective mechanism in Vichy syndrome, it had been non-specifically implicated in a very wide range of conditions, particularly conditions affecting post-mitotic tissues, such as the cardiovascular system, such as the central nervous system, and the musculoskeletal system, but it also had, for example, been implicated as an important mechanism in cancer biology. And this important role of autophagy was confirmed in Vichy syndrome in EPG5 mutated patients, which we could characterize as one of the most extensive multisystem disorders in humans. I've highlighted in red the key diagnostic features of the condition, in blue other neurological features. But as illustrated by the features highlighted in black, virtually any organ system can be affected in this condition, emphasizing the importance of the gene involved and of the pathway involved in this very rare condition. And we have by now identified more than 200 patients with mutations in this gene. And bearing in mind that when I met my first patients, there were only 8 patients known, we noticed something which you actually see quite often in rare disease research, that once you have found the gene, you will find that the range of problems associated with the gene is actually much, much wider than what has been initially associated with the initially described rare condition. And we have now seen an extension of a clinical problem into much more common clinical features, for example, epilepsy, movement disorders, including Parkinson's disease and also neurodegeneration. And that is something which is also consistent with disease manifestations in heterozygous carriers of this condition, which may develop cancer and which may develop movement disorders, in particular Parkinson's disease. And we have also tried to elucidate the molecular basis of these conditions. We have relevant animal models in mouse and fly, which replicate the disease phenotype. We have also conducted metabolic studies identifying potential targets for pharmacological modifications. And we are also working on RNA-based gene therapies, which will, however, benefit only a selected number of patients. And although I have used the rare condition Vichy syndrome as an example, that is of course the approach which can be translated to other rare diseases, that you should take a structured approach, try to understand the gene defect, the molecular basis, generate relevant animal models where you can then test orphan or repurposed drugs. And the last part of my talk, another observation which is common in rare diseases is once you have found one rare disease, you will actually find that there may actually be a much wider number of conditions associated with the same genetic defect. We have now identified almost 50 conditions which are linked in defects in autophagy. And these conditions are often related to genes which affect certain parts of the pathway, in particular the fusion between the autophagosome and the lysosomes, meaning that if you find a drug which can treat one of these conditions, the drug is also likely to benefit other conditions. And these conditions have very characteristic, recognizable clinical features, and they often have a biphasic course linking neurodevelopmental to neurodegenerative features. And I think that is a very important aspect because these conditions, in my mind, represent the early onset part of a spectrum, which on the other end results in much more common conditions such as Parkinson's disease, ALS, or dementia. And I think this is important to bear in mind, because autophagy essentially is a mechanism which counteracts the effects of normal aging. And any treatment which benefits this rare condition is likely to benefit more common conditions, but also in particular age-related conditions. And just to highlight that autophagy is an eminently pharmacologically modifiable pathway, there are a large number of drugs which can both enhance and block autophagy with huge potential for therapeutic interventions. And I want to finish here, and I think I want to show my last slide, which is one of my favorite slides when I talk about rare conditions, and that is the tale of 6 blind men describing an elephant. And I think that is a situation we are often in when we approach rare diseases, because often we approach rare diseases from a highly specialist perspective. And I think it is very important to bear the wider picture in mind, and in particular to bear in mind that rare diseases affect very common and very important biological mechanisms. That is often the reason they are rare, and that these mechanisms are actually implicated in much more common conditions, and that therefore any drug development for rare diseases may benefit much more common conditions. And I want to thank here a large number of colleagues, collaborators, and funders. I want to thank Beacon for inviting me here. And I also emphasize one point I didn't make in my talk, the importance of really having effective international collaborations when working on rare diseases, and also to work very closely with patients and patient organization. A lot of my work has been supported by patient organization. Actually, the mouse model we have was funded through the support of a father whose son has Vichy syndrome. And as I mentioned in my comment earlier, I've also always felt when learning about this condition, but also about other rare conditions, that I've learned as much from the patients and their families as they have learned, hopefully, also from me. Thank you.